Hi everyone, my name is Eliza Clark and I'm going to talk for a couple minutes today about how rapid adaptation to day length affects dispersal evolution during the range expansion of a biological control agent. So evolutionary theory makes some predictions about how populations will evolve during range expansions. First, we predict that dispersal ability will increase at the edge of the range expansion. And second, that local adaptation will occur during range expansions to heterogeneous environments. Increased dispersal ability at the edge of a range expansion evolves due to spatial sorting of dispersal ability. To illustrate this, imagine a population like depicted at the left here that shows the population expanding to the right. The good dispersers in green will move further than the blue dots, and over several generations of this and with assortative mating between the good dispersers, there will be an evolution of increased dispersal ability at the edge of a range expansion. Most range expansions occur across heterogeneous environments. For example, summer day length increases at increasing latitudes. Because day length is used as a seasonal cue, a cue for seasonal change, the fitness of individuals increases when individuals are in sync and adapted to the day length that they experience at their local latitude. So our main question for this research was about how strong local adaptation to seasonal cues like day length impacts dispersal evolution during range expansion. And we use a biological control system to study this. These systems are really well suited to studies of range expansion because we know a lot about the history of the releases of the biological control agents. Our system is with tamarisk, which is an invasive tree in Western North America, and the tamarisk leaf beetle, Diarrhea carinulata, which was intentionally released to help control tamarisk. It does this by eating the leaves of tamarisk, which slows the growth rate of the invasive tree. The beetle was released into the United States starting in about 2001, and it was able to establish really well at sites that were above the 38th parallel, but the releases that were below the 38th parallel largely failed. However, in about the past 10 years or so, the beetle has been able to expand their range southward, mostly doing this by themselves. So first we wanted to compare populations at the core and edge of the range to determine if there was an evolution of increased dispersal ability at the edge of the range as predicted by theory. So we collected populations from the core of the range, which are these northern sites, original release points in blue, and we collected 4 populations that were at the edge of the expansion in 2017 when we collected these beetles. We took all of these beetles back to the lab and then there performed flight trials on flight mills to determine the dispersal ability of individuals using flight parameters like how far and how fast each individual beetle could fly. I'm going to show you the results for 3 flight parameters: distance, number of flights, and average flight speed. And as you can see, there's a lot of variation between individuals and how far and how fast they can fly. However, there's no significant difference in any of these flight parameters between core and edge populations. So despite those strong theoretical predictions of the evolution of increased dispersal ability at the edge, we do not see that in this range expansion of the tamarisk leaf beetle. One main assumption of that theory, that increased dispersal ability at the edge, is that individuals are expanding across homogeneous environments. But as I've already mentioned, we know that's really not the case for many real-world range expansions. So summer day length is one of those non-homogeneous environmental factors, and it's often used by species to time critical life events. So we wanted to investigate how local adaptation to day length was possibly impacting that evolution of dispersal ability. To understand better how day lengths impact these beetles, we need to talk about diapause. Diapause is basically insect hibernation, and it's a process during which insects will stop reproducing and prepare for harsh conditions like cold temperatures in the winter. And I'm representing diapause in this beetle as this little beetle here in a blanket, and this will come up later as well. So these beetles use the day length to know when winter is going to come. And at the northern sites, they use a day length of around 14 hours, which occurs during late August, and that's when they know when to prepare for winter. But if the beetles use that same day length cue of 14 hours in the south, it occurs much earlier in the year, in about June. So when beetles were originally released at these sites below the 30th parallel, they would get ready for winter at the 14-hour cue, but then it was June in Arizona and they experienced really high mortality, as you might be able to imagine. So one way that researchers have been able to study adaptation to day length and diapause timing is through a trait called critical day length. So in one study from our collaborators I'm showing here, these researchers found that 4 populations of the beetle had significantly reduced their critical day length in the space of about 5 years. This translated into longer reproduction in the summer because they were better timed with the day length that they experienced at their latitude. Critical day length, though, is by definition when 50% of a population is in diapause. So this is a population level measurement, which means one number for one population. And while this has been really useful in studying diapause timing, we wanted to use an individual level measurement to better understand the variation between individuals and also the genetic basis of the trait. So we defined our own trait called days until diapause. This is the number of days it takes a single individual to enter diapause after we switch that beetle into a new day length. So we wanted to know some things about this new trait that we defined. And first we needed to know whether days until diapause can respond to selection. So the response to selection depends on 2 factors, which are the strength of selection and heritability, or H squared. So we needed to determine the heritability of days until diapause, which is how much variation in the trait is controlled by genetics. Because even if the strength of selection is strong, the trait can't respond if there is no heritability in the trait. So secondly, we were interested in knowing how the environment that is experienced at different points along the range expansion would impact heritability. We studied heritability in one population from the north using a paternal half-sibling breeding design. And this is basically where one male is mated to multiple females, and then we measure days until diapause on all of their offspring, which are half-siblings. And we do this many times. So each of the half-sibling families got split into 2 treatments here. One was a long day treatment, which was a day length of 25 minutes, less than the critical day length of the population, which is about 2 weeks later than that day length would occur in the field. And a short day treatment, which was about 4 weeks later than the critical day length in the field. So this plot shows the mean days until diapause for each half-sibling family. And these are just in order from least to greatest to help with visualizing this trait. So our results show a lot of variation between families in the number of days it took them to start diapausing in the home environment. So it took families between 5 and about 25 days to enter diapause at the home environment. And all of this variation resulted in a very high heritability of 0.7, which indicates that there can be a strong response to selection when the beetles are near their home environment. However, when we looked in away environments, it was a very different story. There was a lot less variation, and this resulted in no significant heritability for the trait, which means that days until diapause cannot respond to selection when they are far away from their home environment. Next, we wanted to determine if days until diapause showed a signal of local adaptation across the range. We used the same 8 populations that we collected for the dispersal experiment, and we did a reciprocal transplant with a northern and southern environment, where each environment was a day length that was the critical day length in either the north or the south. And to remind you, the critical day length is when 50% of an adapted population is in diapause. So I'm going to show my results in plots that look like this one. So I'm going to explain one here, and then I'll show you the rest on the next slide. So to determine whether beetles were in diapause, we looked at whether they were laying eggs. So when they were laying eggs, they were not in diapause, and when they stopped, we knew they had entered diapause. On day 0 of the experiment is when we put the beetles into the day length treatments, And on day 43, we ended the experiment because this gives them plenty of time to respond to their environment. Each line on the graph represents a single individual here. So they all start out at the top, not in diapause. Then on the day that the beetle stops laying eggs, they drop down into the yes diapause category. Some beetles never diapause though, and their lines will stay at the top the entire time. Okay, now for the results of the reciprocal transplant. When we put northern beetles into the northern environment, we found that about half of them entered diapause, which, because they were at their critical day length, indicates that they were adapted. They got ready for winter, and then winter came. However, when those same northern beetles were put into the southern day length, they all entered diapause really quickly, which would be like entering diapause way too early in the year. So when southern beetles were put into a northern environment, we found that they were also mismatched, but for a different reason. Most of them did not enter diapause when that would have been beneficial in the northern environment. Finally, southern beetles in a southern environment were well adapted. About half entered diapause, and so they were well timed with their environment and entered diapause in time to prepare for winter in the south. So together, these results show a strong signal of local adaptation because fitness is highest in their home environments. So to go back to the original question about how local adaptation to seasonal cues will impact dispersal evolution. So first of all, we found that adaptation of diapause timing has been really critical in allowing the establishment of dirabda at those southern locations. Furthermore, our heritability results suggest that the movement of dirabda southwards needs to be slow enough so that it gives the beetles time to respond to selection in days until diapause, because they can't respond to selection when heritability is really low, far away from their home environment. And finally, we think that the low heritability of days until diapause away from home could be preventing spatial sorting from occurring, which could explain why we didn't see an evolution of increased dispersal ability at the edge of the range. So to broaden out these findings a little bit more to the study of range expansions in general, we suggest that multiple traits should be looked at when studying range expansions, because as we've seen, adaptation of one trait could impede adaptation of another trait. And finally, we propose that the theoretical predictions of increased dispersal ability at the edge may be restricted to only homogeneous environments. In heterogeneous environments, selection and local adaptation could be more important and impede spatial sorting of dispersal ability. As range expansions become increasingly common in our era of global change, it becomes more and more important to accurately understand the dynamics of range expansions and predicting their rate of spread. Our findings can help us understand better how evolution will impact range expansions into the future. And with that, I'd like to thank all of my undergraduate helpers and of course my lab group. And I'd also like to thank my department and program and the USDA for their funding and support. And I would love to answer any questions you have in the discussion below. Thank you.